All right, let's talk about the chambers of the eye. There's really two, the anterior chamber and the posterior chamber. I'm going to flip to the picture for a moment so you can see what those are. All right, the anterior chamber is this portion right here in front of the lens. The posterior chamber is here behind the lens. The anterior chamber contains something we call aqueous humor, and aqueous just refers to very watery. So it does look very watery. Behind the lens, this is the vitreous humor. It's a little bit more gelatin-like, has a bit more consistency to keep the eyeball in a good shape and a good position. So when you actually do dissections of the eyeball, it looks almost like a grape that comes out a little bit squishy. The aqueous humor does circulate throughout, and when it does get to be too much fluid in that front of that eye, that is what we call glaucoma. It is fairly serious to have too much pressure against that front portion of that eyeball. It can create some problems with vision. All right, our last layer of our eyeball is our retina. So that's number 3. It lines the very inner portion of the eye. All right, so it is the inside section. Looks yellow here on this lovely little picture. All right, and there is this little divot in the back, and if you look at it, you can see this little section right above that optic nerve where it takes a little dip in, and we call that the fovea centralis. The fovea centralis is really the portion where we see all of our vision. Okay, we're going to find most of our sensors for our vision right in that center. And when we look at how light should come into the eye, all of the light should be focused with the lens, that one little pinpoint spot, that fovea centralis in the back. And if it is, everything is 20/20 vision. And when it doesn't focus exactly at that one little pinpoint spot, that's where we start to see some differences, like we looked at last semester with our different eye charts. All right, there are really two types of receptors in that fovea centralis, rods and cones, which is what you're seeing here. Okay, so rods have this little purplish appearance, and cones have this more pink appearance in this picture, but they're both photoreceptors, and so they help with vision. This is just what it looks like underneath an actual microscope, okay, in the retina, that inner layer. All right, the optic disc, okay, is just to the inside, and it's right where that optic nerve comes into the back of the eye. It leaves this sort of little empty space that's visible. It is also called a blind spot because there are no sensors right there. And if you go into YouTube, you're just interested in this, you can do all sorts of blind spot testing just to see how large that spot is for you. We did just talk about the vitreous humor, okay, that forms towards the back. And again, it helps to maintain the shape of the eyes. Okay, so here's a nice little peek. All right, so light refraction. Okay, so when light waves come in, they really have to be focused, and that's what that refraction refers to. And so that's why that lens and the ciliary body, that muscle, okay, pulls that lens into different sort of configurations so that we can get all that light focused directly to the back. And if the eye shape is totally normal, you're going to see directly towards the back of the eye. So let's take a peek at what that looks like. All right, so here's all the light, okay, waves coming from the outside. It hits this lens, and what it should do is put that little pinpoint dot at the back of that fovea centralis, and then we see at 20/20 vision. All right, those rods and those cones are found right in that fovea centralis, where the biggest concentration are right there. And cones, okay, really help us with colored vision. And I always remember the C with the C, right? Cones for color. And rods give us more of our black and white sort of vision and our different shades of gray. So they both do sort of different types of things, okay, towards the back of the eye. All right, so here's a nice little look at rods versus cones. All right, there's a rod versus a cone. So you can see the rods are more purple and the cones are more that pinkish color in this picture. All right, again, cones enable us to see in color. To see something in detail, we do move our eyes. Again, that lens moves to focus the light perfectly against that fovea centralis. Rods and cones do contain pigments, which is what we see for different colors. I'm not going to test any particular pigment color, okay, for you guys on the test. But some of the pigments include rhodopsin, which is a purple, opsin, retinal, vitamin A. Okay, so there are just different types of pigment towards the back of the eye, and if you're missing one of those, that's where we start to see color blindness. So if you're missing some of these pigments that are being produced, okay, that's why we see different types of color blindnesses. All right, again, those pigments are broken down very quickly so that when light hits our eye again, we're able to see, okay, and get another action potential, okay, down into our eye and then back towards the optic nerve. All right, again, light-sensitive pigments they're proteins. Again, there's the 3 different sorts. I'm not going to test the 3 different sorts. And it's really just wavelengths that determine which one of those pigments will be released, which is why we see the 3 different colors, red, greens, and blues. All right, again, just how we stimulate those rods, how we stimulate those pigments, okay, gives us different forms of black and white, shades of gray, the colors, okay, and of course, if you're missing some of those pigments, okay, it results in color blindness. All right, the visual pathways, I'm not going to get super detailed, but I do want you to recognize this from this picture, that stuff from your right eye, okay, does cross, okay, over and interpreted towards the occipital lobe of the left, and the left eye crosses over, most of it does, and is interpreted by the opposite side of the brain, so there is some cross. You should also notice that most of your vision is actually done by both eyes, so that when you close one eye, you actually see a good section of what you see with the other. So there's a lot of crossover so that if you do lose some vision, okay, the other eye will sort of pick up for it. The only thing you would possibly lose is some of this peripheral vision out on the side, okay, which is only done by either the left eye would see out in the left periphery, and the right eye, which is the right periphery.